to me to have these charts with us as a keynote speaker. I was wondering how a small boy like me can be a keynote speaker. You guys here, you are better than some of us. You have experience, you are vast, and you are more skilled. But collaboration is the key. Working together is the key at any sphere of life. Nobody knows it all. Nobody is the best. And that is why we need to leverage on our strengths and our weaknesses so that we can be better off in life. And if the aim is to better the continent, the aim is to better our earth system, agricultural environment. So we have no room for competition than collaboration. So I don't know if it is possible for me to share my screen. Perfect.
Yes, you can, sir.
Okay. So I can run through. Great. Yeah. Yeah. I believe everybody can see my screen now.
Yes, we can.
Great. So I was given a very big topic. It has to do from data to discovery at scale. AI, open genomics, data equity for global bioeconomy. Oh my God. I think when I heard about bioeconomy, it got into my mind what I've been working on for the past 6 months. You have to get yourself to where we are going to. I have been browsing and chatting and investigating the most worthy scientists in the world, and I can see some of them. It's only that they are silent. You don't know that. So people don't know that scientists can get to the list of most worthiest people in the world. If after this training or any other thing, you can check online. They exist, and they are doing fine, doing fantastically well. But in the school, most of the training we have doesn't do anything about economy. I either train a student to be a co-lecturer like me, or I train a student to be a researcher, not teaching the student about the business aspect of it. And that is what bioeconomy is about. How can you drive good economy from biological processes rather than getting PayPal out? It's good to get PayPal. It's good to have the knowledge too. But when you have the cash too, you do that work at another scale rather than stressing yourself doing that. So at least celebrating 5 years of Biosphere, contribution to bioinformatics, data science. These are things that it's worth it, and innovation. And I know Biosphere was bearing it in before we move on to name. So the global bioeconomy is reshaped now by AI and machine learning, open genomic data, and high throughput biology, then equitable access to data and innovation. Today, how can Africa and Global South lead this future? That's the question for you. You can think deeply about that. So this topic matters because the world is shifting into data-driven biology. We have AI tools out there to discover vaccines, drugs used in agriculture and public health. Genomics is becoming more accessible, but still unequally distributed. There are some areas that things were expensive, but now people can still get it affordable now. We have some labs in Africa that are having the sequencers, having those skills, those facilities. It's different from what happened the year past. So low, medium, and income countries risk being data consumer instead of data producers. So we consume the end product from the data rather than being the one producing it. So we need to build an inclusive global economy with shared access, shared benefit, and shared capacity. You see what I say? Build an inclusive global economy with shared access, shared benefit, and shared capacity. That is the way to go. And the era of biological big data The sources of data is NGS, multi-omics platform, electronic health record, real-time pathogen surveillance, environmental and One Health data. So the challenge we have now in Africa is how do we turn this overwhelming data into meaningful discoveries? How do we bring out products from this data set that we are generating? So AI has been a catalyst for discovery. In an example of AI is transforming biology. We have protein prediction. You can be talking about AlphaFold, MetaFold, ITASA. We have a lot of them like that. We have disease outbreak forecasting. There are a lot of tools that can help you forecast the disease outbreak. We have drugs and vaccine design, genome annotation, precision medicine. We have high throughput screening analysis. So AI gives us speed plus scale plus precision, and that is where AI is coming in. Then, what is open genomics? That is benefit of open genomic data. It allows to have a faster outbreak response, collaborative global science, new drug and diagnostic development, and capacity building across border. And I was talking about GSAID, NCBI. These are platforms that is open, that you have access to data. But there is a gap. That's data inequity. What are the key barriers? Limited sequence infrastructure. So you can't compare the number of data generated in Africa with other continents. Then low participation in global data set. There are a lot of things that are done in precision medicine, reference genome, every other thing, mapping disease. But what's the population of Black population that is there? I'm not talking about Black America, but the really Black people that are there. We have restricted access to tools and computer resources. How many of our lab have remote server, have those high computing power machine that can be? So funding gap and limited trained bioinformaticians. I'm sincere with you. We have limited trained bioinformaticians in Africa, in Nigeria. It's not somebody that knows the introductory part of it, not somebody that can use it too, but somebody that have a deeper knowledge of interpreting the information, the output of the analysis. Then ethical concerns about extraction of data without benefit sharing. We can do the project, share data with some guys outside the country, and lead to product development. How do we share the benefit? How do we share income? So LMIC researchers often excluded from breakthrough they contributed samples to. So you see that they will not be acknowledging them or querying them or even at the front there to talk unless you are just at the back door. So how do you build a bioeconomy? A thriving global bioeconomy requires accessible data, democratized AI tool, sustainable infrastructure, strong policy on equitable data sharing, local manufacturing and innovation, South-South and South-North consortia. So we must move from users to creator to exporter of biological innovation. And that is been my space for the past 6 years that I've been working in seeing not just getting data and analyzing data. How can we get product out of this data? We are not there yet, but we are closer than where we were some years back. So, and that is what we want to do, because most of the biological innovations that is happening in Africa, they are all imported innovation, not what is locally driven by our local information. And let's just see our population. If you can do that and see a lot of people suffering and say how much money can you make from this? So look at the case study of African Genome Leap. Look at what happened in the era of COVID-19 and look at what Africans were able to do. Then look at Rising Genomic Hub. It was agency before, it's now an institute. Look at A3 Africa. From A3 Africa now we have ABI, that's African Bioinformatics Institute by Professor Nicola. Look at what Helix is doing. Look at what HackBio is doing. Look at what Genomic Hub is doing. So we have lots of genomic hubs rising up. We have regional sequencing network, and we have growing AI talent pipeline. So Africa can shape global discovery if this investment continue scale. So what are the opportunities in AI plus genomics and equity? There will be rapid detection of emerging pathogens. So pathogen can reliably cause problems for us. We detect homegrown vaccines and diagnostics. Look at how long it took them to develop malaria vaccine. Now we are still battling with Lassa in Africa, in West Africa. Where is it? Look at African swine fever that is killing our pig. No vaccine yet. You can see we can lead to precision agriculture and biotech startup. Though that will create new jobs and biotech entrepreneurship. And like I do tell people, what happened to financial states, financial ecosystem like Paystar, Flutterway can also happen in biology. So it is we that we know how to come together, think through, and see how we can do that. So there will be competitive African bioeconomy that feeding global innovation. So what are the framework for the action? One, invest in genomic capacity, and that's what you guys are doing, which we are doing it, sequencing up, training centers, cloud computing assets. Then we adopt open science principle. That is fair data, share repositories, and a good ethical frameworks to be on ground. Then another thing is advanced AI literacy and research. So we need regional AI bioinformatics program that either a AI cloud-based bioinformatics courses and open source ML machine learning model for local pathogens. Like I was looking at some guys yesterday, they have to do some other work on Nesclid, on influenza A. They added something to it so that people can use it for genotyping, contributing to tools. Then build public-private innovation ecosystem. That is biotechnology startups, Earth Technology Accelerator, then industrial academia partnership, so that there will be a synergy between the industry and the academic. So what's the role of Biosphere today as you are celebrating your 5 years? Now, in the next decade, create African-centric open data sets, power research with accessible AI platform, train the next generation on genomics innovators, establish multi-country collaboration, then drive policy dialogue on data governance. So Biosphere will be a catalyst for global equitable biotechnology growth. So vision for the future. Imagine a world where African data set train global AI model. And that is why when it comes to precision medicine, there are a lot of things we cannot agree to, because most of the data, the model, there are not enough data from Africa to train that AI model during the machine learning process, because the more data, robust data you have, the better the precision of the result that you have. Then diagnostic and vaccine are co-developed across continents. We join hands together. Young scientists lead world-class discovery pipeline. Knowledge flows in all directions, not in a secluded place, not within a group, but in all directions. Then innovation is inclusive and global. This is the future the bioeconomy needs, and Africa must help shape it. So my final message to do is that data become powerful when it is open, ethical, equitable, empowering to all communities, from data to discovery at scale. So let's build a global bioeconomy that includes everyone. Thank you for listening, and I'm glad that we are in for today and tomorrow to do much and enjoy ourselves in this space. Thank you so much.
Awesome, awesome. Thank you so much, Dr. Oladipo. Please let's give it up for Dr. Oladipo. More. Thank you so much. Thank you for everyone that responded. Yeah, so thank you so much for that presentation. So succinct and straight to the point. Yeah, generally we like you mentioned at the beginning of the presentation that we had always seen research as a thing of, okay, let's just add knowledge or let's just add to the body of knowledge, right? But we are not looking at the economic aspect of it, because we can actually make some money and also improve the economy of the country by the result of our research, which is great. So I like the fact that we are now shifting attention, of shifting focus, and also focusing also on the part of the economic on the part of the finance or financial aspect of research, and how we can scale from the lab to probably a company, a biotech company, and we can see what these companies that you mentioned, these startups that you mentioned, including Helix Biogen Institute, right, is doing. Awesome. And please, if you have any question, you can just drop it in the chat. And if you want to ask, you can raise your hand and I would call you to ask your question. Yeah, but I will go first. Um, At some point, you said a thriving global bioeconomy requires accessible data. But now we see that, for instance, even in Africa, you mentioned it at some point also, that we don't document stuff. We don't have a good repository of data from Africa. And we need accessible data. And even people that have the data, there's a place of hoarding data, right? I got to know that even big biotech or pharma companies, they have their own kind of chat GPT where because they want to ask information, they want to make some inquiries and all of that, but they don't want information about the company to leak out. So they create their own model within the company. So that means that whatever data they are sharing in there is not available to people outside. And this data could be useful for people outside. So there's a place of uh, unavailability of data, especially or insufficient data in Africa, and the place of data hoarding too by countries or even company. But we need accessible data to have a thriving global bioeconomy. So if this continue, is there a future? Is there a possibility of us achieving this aim?
Yeah, I think let me talk from business point of view. Some of the time we talk much of academia that I have a data like pull it out. In business, it's business. Business is business, and the essence of business is to make profits. So, and there's something we call IP, intellectual property, and something we call patent. So there is no company that will release out the secret, because the releasing of the secret of that company means that that is the doom, the end line of that company, and there are laws that guide that protect that. So what I would have suggested is that look at NCBI is owned by US, EBI is owned by UK, DDJB is owned by Japan. Do you know that China has their own separate databases?
Yes.
And you don't register there, you can't have access to it. So it is left for Africa to come and say, oh, some people have applied for grants, and let's build an online repository where we can keep our own data set. And if you need to use our data set, there will be terms and conditions. Not that it's going to be open. You can have access to it, but there will be terms and conditions attached to it. For instance, I was telling some of my colleagues that, oh, when we are doing human genome sequence, what if in my village I got a whole genome sequence of a woman or a man, and that leads to discovery of a drug, but that discovery was done in US by one big pharmaceutical company. I said, if that drug is out and the person will not know that his own genomic data set that led to that discovery, can he or she afford the price? And if everybody knows that, yeah, I couldn't remember the story now. There was a lady, a family that sued a pharmaceutical company because they used the data to develop drug. So he requested for royalty, a good sum of millions of dollars, because they have made enough profit for it. So what I would suggest is that we take responsibility of our lives. We take responsibility of our data set, and we ensure that we use the data set. Even submit on NCBI, they can tell you when you want the data set to be released out for people to make it open. Not that you're not making it open. I do tell my students, if you have not done like 60 or 70% analysis on your sequence data, we don't submit. Because the moment you submit, others will pick it up. I work on vaccine design and development. Initially, when we do the vaccine, shell knows, we put the construct sequence in the publication. But later I got to know that some guys in China already set up some mechanism. They are working in the vaccine industry. So if you publish today and you put all those sequences, they take it from you and start expression and start experimenting with it. And they might be lucky that that's what is in the market. But the person that put it there in his publication is sitting there in his lab in Nigeria and is suffering from fund. And some people are making billions of naira from it. So when it comes to data, we must have a knowledge how to manage our data, how to put actually it's open science, but there is a way you have to protect what is open so that we can benefit from what is open. We have to practice
Okay.
open science, but we have to have a structure, a policy in place to protect our data.
Awesome. Structure and policy for data protection. Thank you so much. Let's just applaud our Dr. Oladipo once again. Thank you for coming and giving the keynote speech. So we'll be moving on to the next item on the agenda, and that is a welcome address from one of the co-founders. So give it up for Martin as he gives us the welcome address, and we start the first day of our 50th anniversary program.
Okay, thank you very much, Ololade, and good evening, good morning, good afternoon to everyone. My name is Martin Ikhoyameh, and I want to welcome everyone to our 5th anniversary in Biospherea. So I want to begin with the words of Chino Achebe. He said, "When we gather together in the moonlit village ground, it is not because of the moon. Everyone can see it from their own homes. We come together because it is good for kinsmen to do so. Likewise, we have gathered here today not because as individuals we cannot pursue our passions independently, but rather because it is good, pleasing, it's fulfilling, even rewarding to connect with other people. Yes, the importance of community cannot be exaggerated. There is always a need and desire to bond with others. Biospherea began 5 years ago as Science Cafe, a small group of 12 individuals, shortly after the pandemic, which, as most of us know, is one of the most physically and socially isolating period of our lifetime. And at that time, wealth was the only one I knew as a friend. But now I think everyone knows that the story is different. I've met a lot of wonderful people through the community. So today it is a great honor to be here as we celebrate 5 years of our community's journey. We have grown and continue to grow as a strong community where ideas are shared and refined, where skills are honed and nurtured, a place where we work to equip and support ourselves and other Africans in our own little way to be able to confidently explore science careers and passions. So much has been achieved already from this community, from our workshops on transferable skills to article publications and hundreds of engaging sessions on different topics in biosciences, including open discussions. We have certainly come a long, long way. The theme of this anniversary is AI-driven bioscience, from data to discovery. Just like the keynote speaker mentioned, it offers wonderful opportunities. And this anniversary's theme reflects our commitment to helping our members and the broader African Bioscience Society to stay current with trends in the global scientific arena. In today's world, massive data sets can be rapidly analyzed and translated into meaningful insights, which gives scientists amazing tools to solve problems across many disciplines. AI tools should not be viewed as replacing us in our role as research scientists or as professionals. Rather, they should be seen and used as opportunities for amplifying our efforts. They should be viewed as a means of tackling problems in ways not possible even 10 years ago. So, whether you are a student just gaining exposure to science and research or already a professional in the field, it is important to acknowledge that it's a wise thing for everyone to make the most of available resources. And what better way to increase efficiency in what we do with minimal effort, but to make use of the right tools such as AI in our time today. So with that, I want to once again thank everyone for coming. Thank you to all our members for continuing to believe in our mission and contributing their skills, time, and ideas. Thank you to everyone joining this event, and to all our speakers, the keynote speaker and those that will still come afterwards. It is my hope that this event will inspire new ideas, connections, and opportunities. So I would like to leave us with the words of Ralph Waldo Emerson, which says, "If we are related, we will meet." So I'm glad that this opportunity has come for us to meet, as we all have a shared passion of contributing to our society and building our lives. Welcome once again. Happy anniversary, and thank you, everyone.
Amazing. Thank you so much, Martin. That was really powerful. I really enjoyed it, especially the proverbs. All right, so we'll be moving to the next our next session, which will be by Michael Olufemi, and he'll be presenting on the topic AI agent for autonomous space crop production. Michael Olufemi is a fourth-year PhD candidate in applied biology at the University of Massachusetts, Lowell, where he studies evolutionary genomics and data methylation, and he has a focus on gene copy number variation. Beyond his dissertation, he also contributes to NASA's Analysis Working Group, which is AWG, on space biology projects, and is deeply engaged in hackathons where he enjoys solving real-world problems and designing data-driven computational approaches to impactful public policy. With a round of applause, let's welcome Michael Olufemi. You're welcome. All right, you can you're welcome, Michael. You can unmute and start. Thank you. And please feel free to drop in your comments, and if you have questions in the chat, we're going to pick that up. And I believe maybe whoever is directed at to can try to maybe answer in the chat
Yeah, well,
also.
thank you very much. Yeah, I was unable to unmute myself, so but
Okay.
okay. So good evening, everyone. Good day, wherever you are. And it's a privilege to be able to present in this year's anniversary. So I'll be go ahead and share my screen and just get started with it. Okay. So just trying to see. So is my screen visible?
Yes,
Yeah,
we
it
can
is.
see your screen.
Yeah. So welcome everyone. My name is Michael, and I'm a PhD student at the University of Massachusetts in Lowell. And this is just some of the project I did over like this summer at NASA Gen Lab. And we know like the buzzword in the past 1 year, 2 years has been AI, and there's always AI for everything. And as the topic of this anniversary is, this theme is AI-driven bioscience from data to discovery. So what can we do with this huge biological data that we have? So I'm thinking about how we can use AI agents for a simulated plant lunar habitats. So this work was not done by myself alone. So I had a team of interns that we all worked together. We were both interns at NASA Gen Lab over the summer, and there's Wale, which will be taking the technical section later to get today. Anu and Oluwaseun. Those are my friends that we worked on this together. But we're presenting this to you guys. So when it comes to space research, every form of space research, from the word space, means it is being done outside um, Earth. So you have to go on a space mission, you have to plan, and each space mission generates massive volumes of data. And usually they are exceeding the capacity to analyze them real time. So you most of the time you wait for a particular mission that can take 180 days, 200 days for the whole mission to be completed. Then you have data being transferred back. Then it's after like 200 days that you're able to make sense of what has gone on in the mission. So, and also even if you try to analyze real time, some of the real time data, there's usually communication delays with it, since the whole communication system is not as robust as they want it to be, and even though there's improvement and there's work around it. However, making on-site decision is essential for this mission success and safety. And we thought that AI is one of the keys to unlocking this autonomy in these critical space systems. And so we took inspiration from Earth, because on Earth you already have self-driving labs, you already have self-automated labs where scientists just plan the experiment and they give it to like the robots and the automated labs that they have, automated systems. So this system can make everything, sometimes from DNA extraction to sequencing. It can do all this analysis itself, barcoding, different stuff. So we know that on Earth, we can make all these self-driving labs already. So this already gives us inspiration that this is possible for us to do it in space. So the goal of our project was that we are going to develop a data-driven AI agent that can autonomously manage and sustain a simulated lunar plant habitat. And first, I'm just going to briefly talk about what an AI agent is. So an AI agent is simply an AI that has access to tools and can make decisions by itself sometimes. So an AI agent should be able to perceive its environment, it should be able to make decisions and take actions autonomously. And all this together is to achieve a particular goal. And here you can see an AI agent usually have access to tools like API calls, access to internet, can interpret code, then it can react with its environment. Then those reactions with its environment, the AI agents can take those reactions and put them in memory. The AI agents can delegate tasks. Then having all this together, then it can make autonomous decision. And because this same autonomous decision can also be influenced with human control. So you can decide whether you want it to be 100% autonomous or you also want human control. So that is what an AI agent is. So an AI that has access to tools that can make this set of decisions. So our framework was, we assume, like in space, where Mars or most likely Mars, we're going to have this kind of setup, a plant bed, where we have like a robot, and this robot can take samples from this particular plant. We have camera that takes images. We have fan. We have light. We have all these nutrient reservoirs. We have a cooling device. We have a airflow meter. We have sensors for CO2, sensors for temperature, sensors for pressure, and all these different environmental data sets. You can get them in our lunar pod, and we have a way to log all this environmental data. So the goal is that from this lunar pod, we are going to have an environmental data, we're going to have an image data, and we're going to have genetic data. So we're going to collect samples from this plant in time series, and this robot is going to be connected to like a little sequencer, and this sequencer does some form of metagenomics, does some form of transcriptomics, and we have all this data together. So we have this integrative multi-omics and environmental data that we want to use to make decisions in space. So we know that since this is like a simulated project, so we have data already for all of them. So we have imaging data from Exolab, we have transcriptomics data from OSDR. So these are open source data. Everybody has access to this data. All these data say they are open source data. So we have Eden ISS, we have Apex and Veggie. Apex is the name, they are names of missions of space missions, and Veggies are names of space missions. So we have all these environmental data together that we are going to stream into our AI agents. So we have to define what is the control gap of what our AI agents can do. Our agents can control LED light. So if we need to increase maybe some form of light, the AI can control that. It can control the fan. It can control like the camera. It can control the cooling device. It can adjust for pH, can adjust for water and nutrient pumps. And these AI agents can control omics experiments. So it can sequence whenever it needs to. However, it cannot control for human behavior. We expect that in space. There are times where we have astronauts that will also be there, but you cannot control for this behavior. It cannot manipulate the plant genomics, at least for now. We don't think it can manipulate the plant genomics maybe via gene editing or any stuff. And it cannot control like maybe direct power supply and some external hardware. So we were able to define explicit what are the control gap for these AI agents. So this is how the architecture of the AI agents looks like. So we have this environmental data, which is going to be temperature, CO2, relative humidity, pressure, and we have omics data, which is you sample the plants, take DNA from it, take RNA from it, which is what the robot will be doing in the lab, in the self-driving lab. Extract DNA, extract RNA, sequence with all this automated system that we have. Then we have imaging data, takes images of the plant. How greenish is it? How yellowish is it? And it's going to log all this information into our data logger. And we have this hardware. This hardware is what is linked to this imaging, omics data, and environmental data. And we have our decision maker, and we have a dashboard where we can monitor what is going on. So basically, they are all linked together because the decision maker, which is the AI agent, can influence the hardware. So it can tell the hardware that, the temperature is probably lower than the critical range that you want it to be. Increase the temperature. So the decision maker can make that, which is the backbone of the AI agent. So altogether, the AI agent, the software, can interact with this data and interact with this hardware. And back on Earth, there's a dashboard where you can visualize what's going on. So to deconstruct what the autonomous decision maker looks like. So basically, we have, like I said, real-time data strips, CO2, temperature, relative humidity, RNA images. They all go into our data type classifier. So basically, as it's coming, we can tell, this is CO2 data, this is temperature data, this is an relative humidity data. Then it comes into our agent orchestrator. So this is the backbone of all the agents that we have. So the way we made our agent is a multi-agent or multi-LLM approach, which means we have each agent designed for each different data sets. So we have different so it's a multi-agent system, our AI agent. So we have an agent that is just to monitor the environment. So when it comes to CO2 temperature and relative humidity, they goes to environment, the agents to monitor the environment. Then when it comes to metadata, which is probably information about the plants, species, and this different where was this sampled from and stuff like that. So we have this metadata fetcher. We have RNA-seq analyzer, which also is an agent that can also analyze metagenomics and analyze abundance of bacteria and healthy bacteria and harmful bacteria. We also have an agent that can convert the imaging data to spectral data. And we have an agent that can make the summary. So basically, all this together is going on in one single autonomous decision maker. And we have an anomaly detector. So it summarizes what it's getting from this data stream. So the data stream is going to come, then we have the anomaly detector. So I'm going to detect, so we can set some form of threshold and say, at 30 degrees Celsius, that is the critical or the ambient temperature or the normal temperature we want these plants to function in, based on literature, based on other stuff that we have that we know. Then we can say, if we have something less than 30 degrees Celsius, if we have something greater than 30 degrees Celsius, that's an anomaly. So we created some form of statistics. So we created some form of statistics, an algorithm to detect this anomaly. And it can detect anomaly over a long period of time. So every 5 minutes, it streams the temperature data. So all the environmental data are streamed every 5 minutes. So that's a lot of data points over, let's say, 3 months. So every 5 minutes for 3 months is a lot of data points, which is like 5 minutes is probably, that is 20 times or is it 20 times or 15 times in 1 hour. Then you do the calculation, like across 3 months, that is a lot of data points for just temperature. Then you have that for relative humidity. So you do all this together. Then we have the system that can control light, control temperature, control water. If the humidity is low, we know what to do. Then we can have log and human feedback. So we have different scenarios that we can use to test this our agent after creating this agent. So I'm going to play a video of how the dashboard looks like and how we can interact with it. So what I'm showing you right now is how the dashboard is going to look like. So you can chat with it. It has an agent decision, an anomaly log, a decision history. Then you can do a sensor visualizer and see how are the sensors doing. So we noticed basically that the CO2 sensors in the pod are picking like CO2 levels of 1,800 ppm. Now, if 1,800 ppm is above what we've done, then the agent is going to think. Then it's going to give his reasoning. It's going to tell, oh, the urgency of 1,800 parts per million is high because the optimal range is 350 to 1,000. So what's going to be the action? It's going to trigger an alarm, then notify the team about these CO2 levels. Then step 2 is going to be like, oh, I'm going to activate a CO2 scrubber to reduce this CO2 concentration in the pod. So all this is going on in this particular agent. So the agent is able to do all this together, make all this decision autonomously without any human interaction. And it can do a follow-up. Because CO2, maybe an increase in CO2 is linked to stress. And this kind of stress can be shown in the image of the plant. Then the agent is going to try to correlate the CO2 data with the image data. And these agents can do that. Also, we're able to also detect relative humidity, which I'm going to skip. I'll go to the last scenario because of time. Here, we're able to see, can we monitor log time plant health and the growth rate via imaging? And so it's going to, we're going to ask the agents. Basically, these are like simulations of what the agents can actually do. And it's like, oh, looks like the image monitoring system in the pod detected is detecting some form of increase in yellow signal, which we want and reduced green signal. So what is it supposed to do? Then the agent is going to know this is the reasoning behind the agent. You don't need to see this once everything is like incorporated with the pod system. This is just most of the reasoning for us to see how is the agent thinking. Is it just giving verbal statements, just like when you ask ChatGPT some kind of questions, then it gives you very verbose response. So we wanted to be sure that these responses are not verbose because the AI agent is also linked to all these open model. So basically the models that we use here is actually ChatGPT too. So we use the OpenAI model. We also use cloud and we use the other open AI system. But basically the AI can make all these decisions by itself. So this is like a simulated project to show that if we can, this AI agent can monitor the plant health, it can make all these decisions by itself, and it can do follow-up. Then after saying, oh, the the yellow and green signal is linked to maybe particular nitrogen level, then it's going to check all these nitrogen levels. It's going to monitor for CO2. It's going to check temperature to see are they also within the optimal range. So it's going to use all these different parameters to make its decision. So, okay, come on. Sorry. So just to conclude, now we are in the new era of AI and autonomy. So if you want to sustain human life in space, it requires a leap of just simple automation to true and intelligent autonomy. And we've demonstrated that our AI agents can already detect, diagnose, and act on complex program within its habitat, blending environmental control with deep biological analysis. It can tell if, based on the greenish stuff, it can also check the metagenomics analysis to see what bacteria do we have in abundance that can cause this yellowish color, and it can make a decision from it. And our future direction is to integrate this into like a real self-driving lab, which is already been done on Earth already, and improve this LLM, have another agent that can judge his decision. Like another way to make ChatGPT smarter is you ask it a particular stuff and you say, "Oh, are you sure?" Then it's going to like probably think more. So if you have another AI agent that can be like a judge of whatever this agent is doing, so it increases the accuracy of the decision because these are all these language model. This is how they work. And with that, I would say thank you guys for listening, and we acknowledge our like mentors during the internship. So yeah, I will take questions now. Yeah.
Yeah.
Awesome.
Thank you so much, Michael. Thank you so much. This is very interesting. And in fact, it's more like I'm getting to know more about what AI agent is and how it applies to bioscience. So do we have questions? Michael is available to take our questions, and we'll just be taking maybe 2 questions. So if we do not select you, please drop it in the chat, and I believe Michael will be able to answer that in the chat too. All right. Okay, so there is a question here. We have a question from Abdul Basit. Okay, we have someone raising their hands. Let me see. Sorry, 1 second. Okay, so Emmanuel, you can unmute your mic, and after Emmanuel and Noro, we have Esther Show. Okay, please unmute your mic and ask your question. 1
Right.
minute, please.
Yeah, good evening,
Thank you.
everyone. Thank you very much for the presentation. Thank you very much, sir, for the presentation. So I just have 2 questions. Firstly, you presented how the agents work and all, how to build intelligent autonomous systems. But I just want to ask if you can also shed more light where humans would be involved in the loop of the workflow, right? Where would humans be involved in the loop of the workflow? Because I also believe there should also be a place where humans are also needed in that. So if you can shed more light on the broader scope of how this system works, including where humans will be in the loop. Also, maybe you can also state more on how this would drive the productivity and also lead to how our goals can be achieved in a much more efficient way. Although, yes, I know, assuming that is definitely why we do this, but if you can shed more light, like give us details on how that is possible so that we can actually see the reason why we should gear towards this trend, right? And then the second question is, you're talking about the fact that you made use of some foundation models like ChatGPT, like that is OpenAI models and cloud. But you were saying something about that they are open AI, but I think those models are proprietary AI models. I don't know if they are open source model. I don't know what you mean by them being open AI. Do you mean that they're open source? Because I think ChatGPT and cloud models are proprietary models are not open source model. What do you mean by that? Maybe you meant something else. I just need more clarification on that, sir. Thank you very much.
Yeah. Thank you. So, yeah, the question was long, but I'll try to answer this last one. Then I go back to the first one. The first one was about where humans can come in. So you can see this chart in the simulation was how can we know the reasoning behind this stuff. So there's a chart, there's where humans can come in. So basically, it does not make those decisions without a log, like a log file. So it makes decision, we know where the stuff is. So humans can feed in, it can receive feedback the same way it can hold in memory the other decision it has made in the past. Like the agents can hold in memory. Humans can also give feedback, and they can also like the agents can act on the human feedback. So it is not a closed loop. There's an human feedback loop in it. Secondly, yes, OpenAI is proprietary. But like when I said open source, that means you can, in a way, retrain it or contribute to it. So they are like paid AI, OpenAI stuff that you can pay for. So like it's not just OpenAI and nobody can use it. That's what I mean by open source. So yeah. So I hope I answer your question.
Yes, sir. Lastly, also more on how this will drive productivity, just so that we have a detailed sense what it brings. Thank you.
Yeah, basically, like you can use this like in labs here on Earth. So most of these challenges I talk about is like delay communication with Earth. The data cannot be analyzed real time. If you're trying to grow a crop in space, and you have to wait 80 days, 90 days until they send the data back to you for you to know what went wrong. But if you know, oh, this plant is turning yellowish, one of the first leaf to turn yellowish, you know that you know what to do, increase a particular set of nutrients before all of them can turn yellow, so you can recover the like the plant's life or something like that. So that is it. That's where you can improve productivity. We can make real time decision because of the distance, from Earth and space. As also you can save money on having astronauts in space every time. So that's how you can increase productivity too.
All right.
All
Thank you.
right.
Thank you very
Thank
much, sir.
you. Yeah, thank you. So quickly, we'll be moving to Esther. Can you just quickly drop a question with like 1 more minute for each person because we're kind of overshooting the time. Thank you.
Okay, thank you very much, Olamide, and thank you very much for that interesting presentation, Michael. My first question is, Michael, was this like a proof of concept, or you guys really use like data from space itself, or you did field experiments with this like from space? Then the second question is that when you mentioned that you guys used open source like ChatGPT and the rest, how are you guys able? I know you mentioned something about the fact that you try to validate that it was not hallucinating, but how are you able to do that validation? Because things like ChatGPT, after a while, they start to hallucinate. Given that you also mentioned that it had data to analyze in like 5 minutes for like 3 months, which is like very large data. So how are you guys able to make sure that it's not really hallucinating after a long period of time? So those are my questions. Thank you.
Yeah. So yeah, to answer your question very fast. The first question, they were real space data. So they were data on those plants that they've been grown in space conditions. So they were space missions. All the data we used were all space missions. So they were data sets from space mission that we used. So all this experiment has been done in space before. So we have all these data set already. So we just use those streaming data sets from space. Secondly, you could tell that it is not, there's a way you set the temperature. So like if you build an agent and you're using, like, you can set the temperature in a way that it does not verbose. So like in the backbone, like in the code, you can set the temperature. Secondly, you can test it repeatedly to know that it's not making, like, you can change it, and their verbal statement is going to make based on like on the logic. We have a format for it which we want it to give its response. So it's going to make those stuff. But based on like repeated stuff, this took like, like 3 months to, we could tell it was not making any verbal stuff. Yeah.
All right, thank you so much. So as a follow-up to that question, just a very quick one. Was the self-driving lab for this experiment in space, or was it space simulated on Earth?
Oh,
And
no.
Abdul Basit asked that same question. So
Oh, no, sorry. So we had like a simulated, we assume like, so we are, what we did was, all these plants, they've been grown in space already. So we have all these data sets from them, all of them at the same time point. So we assumed if we bring them together, it gives this space-like environment. So there was not self-driving lab yet in space. So it was simulated with space data on Earth, basically. Space data, but on Earth. So yeah.
all right, thank you. So quickly your question. That's the last one we'll be taking. Any other question, please drop in the chat, and I believe Michael will be able to answer them in the chat. Thank you.
Thank you very much, Michael. Really amazing presentation. I think the idea of the self-driving labs or automated labs sounds really cool. But my question is kind of a variant of AI hallucinating, because he said that these AI models can monitor CO2 change, monitor so many things about the plant. So are they able to? So I'm wondering if in the design they're able to detect a real kind of a real challenge from like a challenge that isn't real. So for instance, if there's a bug in the system that kind of makes the AI think, oh, the CO2 levels have dropped, are they able to know that, oh, this is not actually the CO2 levels dropping, but this is like a false alarm? So is there provision for the AI model to detect, to differentiate a false alarm from an actual an actual problem that they should take note of?
Well, so that would be an hardware problem. So that means the hardware is incorrectly reading a particular environmental data set. So that would not have to do with the software. However, how are we sure like the software itself can learn? Like most of this stuff we've told it, oh, if the thing is less than 30 or is less than a particular range, so they are not strict values. So if there's a new challenge that is arising, the AI agents can also make decisions, can suggest, like if it's come across something that has not seen before, maybe a very high or maybe very low particular temperature, is going to be able to like flag it. So to answer your question, if it's an hardware problem, it will not be. It's just going to assume the hardware is giving it the correct stuff. But then that will be like then that's when the human feedback system comes in to address that. However, but if it's not an hardware problem, the software itself can learn from its data set and from its memory. That's why there's an AI agent. It can perceive the data, it can hold it into memory, and it can make decision from it. I hope
Awesome,
I was able to
awesome.
answer the
Thank you.
question.
Yeah, thank you so much, Michael. Please let's appreciate Michael one more time. Thank you for that awesome presentation and also the great answers to the questions that came up. So we are going to be moving on to the next thing, and it's our technical session, which will be taken by Adewale Ogunleye, and then he's going to be taking us through the technical experience, his technical experience in bioinformatics over the years, and how to apply the knowledge in our own career endeavors. So just to introduce him, Adewale Ogunleye is a computational biologist and founder of HackBio, a global virtual bioinformatics fellowship that has trained thousands of learners across Africa and beyond in practical project-based genomics and AI for biology skills. With a background in microbiology, getting his BSc and MS in bioinformatics, and his current PhD training in systems biology in Germany. He works at the intersection of biology, data, and automation building pipelines, integrating multi-omics datasets and applying machine learning to biological questions. His experience also includes a NASA Gene Lab internship, where he developed an AI agent for simulated lunar agriculture using multimodal spaceflight datasets. Through HackBio, I beg your pardon, Adewale has shown a strong commitment to democratizing access to bioinformatics training, creating low-barrier, industry-relevant learning paths for students and early career scientists around the world. Please make welcome Adewale Ogunleye. So you have the floor.
Yeah, thanks a lot for I don't know, I don't know who wrote that, but yeah, thanks a lot, and thanks to the organizing committee for inviting me. It's an honor to be here. I see that it's a very large community. I had no idea, and I tried to do some background checks myself. I realized you guys have been around for 5 years, and yeah, it's an awesome job you guys are doing. I believe you should be able to see my screen now. Hopefully. Yeah.
Yes, we
So I know, perfect. Thank you. I know I have 25 minutes. I'll try as much as possible to compress.
can, sir.
So the interesting thing is also that, okay, thank you. So the interesting thing is HackBio is also 5 years, but we are not celebrating. It's my final year of my PhD also, so I'm kind of compressed between a lot of hard things right now. So, but nevertheless, I'm very happy that this kind of community also exists in Nigeria, at least in Africa at large. That is promoting community hard technical yeah, cool. So yeah, I'll be talking about AI and biology. Interestingly, HackBio's theme for next year is AI and bio, actually. AI multiplied by bio. So we are, like he described, we are going to be focusing a little bit, not a little bit actually, to a very large extent on the intersection of artificial intelligence and biology. I don't like to say biotechnology anymore because it's just biology, you know. Biology is more encompassing and it allows you to think beyond borders. Again, my name is Adewale and you can call me Wale for short. I am a PhD student, final year at the University of Tübingen in Germany, and I'm also the founder of HackBio. So I'll be telling you a little bit of our journey as humanity, all of us as humanity in biology in general. Then I'll tell you how HackBio fits into this story. And then I'll tell you how you yourself can fit into this bigger framework. Okay, so I hope that will be interesting for someone. And the point, honestly, is not to come and teach you because I saw that a technical session. I'm like, no, I'm not going to sit down here and do a technical session in 25 minutes. You want to kill me. But rather, I would focus more on inspiring you. Well, now you can, I believe probably there are lots of early career scientists, maybe like me or younger or older, that might be interested in what deep learning has to offer, what artificial intelligence, what can they do outside ChatGPT. All right. Okay. I hope this is your logo. I'm allowed to use it. I just put it there. You guys stick on the copyright. It's not mine. Okay. Okay, so I'm sorry guys, I did not have enough time to create fancy animations on the presentation, so you just pardon me. But yeah, I assume you are not seeing the other parts. But for the first part, what do you think this 010101 means, right? I don't know if anybody can comment on the chat what this code means. Maybe you've seen my presentations in the past and you know what it means. But if somebody can maybe give a clue to what this number means, or this code rather. Yeah, it's a date actually. And the date signifies, no, it's not the beginning of time. No, I'm older than I'm older than that. I'm sorry. I'm no longer Gen Z. I'm sorry. Even though I try to claim Gen Z, I still play football. I still run everywhere. But it's the beginning of the 21st century, actually. It's the beginning of the 21st century. And yes, it's the first day in January in 2001. That's the beginning of the 21st century and the end of the 20th century. And right about this time was when the Human Genome Project was completed, or the first draft. Till today, it's not completed in quotes, but the first working draft was available on around this time. And I think they kind of masterminded it so that it falls around this time, you know. American people are so fancy. Then the next date. Maybe anybody can also tell me what this next date means. Yeah, maybe it's obvious already, but maybe there are so many young people here and you don't know about COVID. But yeah, COVID about 5-6 years ago, COVID was like a thing. Around this time, actually, yeah, face mask anniversary exactly. Yeah, COVID was a real thing, and I got it about 2 or 3 times. I almost died, but yeah, it's a very very terrible sickness, and it's a very tough viral infection. And afterwards, in about 2.5 years later, ChatGPT came on board. But what's the connection between all of this? And I will try to connect everything. So basically, Human Genome Project is the beginning of a revolution in life sciences in general. Either you're in plants, either you're in animals, either you're in microbes. Human Genome Project was a revolution that enabled us to adopt sequencing as a way of life, right? So it started generating DNA sequences, RNA sequences, and we started mining that as a way of understanding life, as a way of understanding and
Oh,
Tony Blair from the United Kingdom. So I don't know if anybody can you hear me?
we lost you for a second, but you're back now. It's fine.
Okay. Sorry about that. So yeah, this was the declaration on I think 26th of June 2000 by Tony Blair and Bill Clinton. And they said, today we are learning the language in which God created life. We are gaining ever more awe for the complexity, the beauty, the wonder of God, God's most divine and sacred gifts, apparently. He said so. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all our lives, and even more on the lives of our children. It revolutionized the diagnosis, prevention, and treatment of most, if not all, human diseases. And this is the key part I held. They said we'll be able to cure all human diseases. And that was since 2000. Unfortunately, I'm sorry to tell you that that has not been the case. We've not solved, I don't think we've even solved any disease. Well, we've solved some disease better than we would have in the past. But so yeah, we are not even close. Since then, we are not even close to solving all possible human diseases. But maybe no, until COVID-19 came, right? And when COVID-19 came, we were able to develop the first vaccine to a brand new disease, in quotes, we developed this solution in less than 2 years. And this was revolutionary. This was like the first time that a vaccine will be developed in such a short span of time. Vaccine development takes a lot of time, takes a lot of effort, takes a lot of commitment, takes a lot of investment. That's financial investment. And yeah, so FDA approves the first COVID-19. And interesting enough is that we achieved this through collaboration from clinics, from pharma, big pharma actually, because the first one was Pfizer and BioNTech. And if you are aware of what the big pharma site is like, Pfizer is like number one, like number one in terms of money, in terms of size, also sometimes maybe in terms of corruption. But yeah, academia and then government. So these people collaborated massively and made it happen. And we've never had this kind of cooperation before. Oh, hello, Miss. Oh my God, somebody's playing with my screen. Okay. Anyways, about 2.5 years later, after COVID came, but then we've developed the first vaccine. Then AI came, right? And AI has been good, has been very efficient since then. I think the first commercial, so I would not exactly AI came around this time, but AI has been around even before then. But the first commercial use case that people could really relate with, sorry, I just, if somebody can help me clear all, yeah, good. Okay, so the first commercial use of AI was really, really ChatGPT. That's what everybody knows. But before then, I think a lot of people have been using AI for image detection, blah, 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 all of these kind of things. So business, in communication, in engineering, in energy sector, name it. I think AI has been really implemented and it's solid over there. But when you come to biology, we have a bigger problem because of what? I'll try to describe the scale of the problem for you. So in Nigerian parlance, I don't know how many Nigerians are here. We use the word learning, like you are a learner, something like that. So AI is still a learner when it comes to biology. There are about 10 raised to the power 30 organisms in the world. Okay, that's the amount of data it has to put in its head. There are just 26 alphabets, and there is a maximum number of words from the dictionary that you can just throw into it. So that's the limit. But for organisms, there is at least 10 raised to the power 30, and we don't know all of them. Okay, not all of them have been resolved. And then for each organism, at least you have 3,000 genes, at least for each organism. And that's massive. So if you multiply or combine 10 raised to the power 30 in 30,000, in 3,000 places, that's some scale. Then all of these genes have transcripts. So genome is different from the transcripts. That is, the DNA is different from the RNA. And then that's also different from the protein. Okay, when you combine all of these scale together, and you are thinking of treatments, so available treatment options, treatments mostly start with chemicals, that's drugs. We have more than 122 million chemicals. And I think this is just the ones that are well documented on PUB10, actually. Some people say it's bigger, it's much more. So we have a massive problem in our hands. No other field in the world, I can tell you for free, has this amount of data that has not been explored. No other field in the world. That's why I say AI is still learning biology. And I think for a very long time, it will continue to learn. So I was reading this manuscript or this article, right? And they kind of gave us a snapshot of what we need to train AI to solve cancer. Basically, to be able to effectively understand AI to understand cancer from beginning to the end across board. This is the amount of data we need. We need 10 to the power 4 cell types and states. So the fact that a cell you understand the cell type at the moment, it's different from its different epigenetic states. Then you have this amount of genes, you have this amount of variants. The variants are the mutations that can possibly happen to all of these cells. You have this gene combination set, you have 10 to the power 4 diseases, you have individuals, 8 billion individuals in the world, antibodies if you are into immunotherapy, and then small molecules. So basically, the idea I'm trying to paint here is that it's so huge, it's so massive, and literally you can't solve it. I mean, we can't finish solving it. Let me put it this way: we can't finish solving it. So, so that's one problem. Another problem is talent, right? And I think this is where you come in into the bigger frame. This is where you come in. Because all of the things that I mentioned above is just data, it's just computing power. I think at some point we can, if the government of the world decides to dedicate full effort into this, I think it can be done. But how will it be done? If there is no human being that is technically gifted to generate this data and then analyze the data. Okay, so it's one thing to be able to generate the data. Let's even assume we've generated the data. Who is going to analyze it? What tools do we have to analyze? And I think the best tool we have right now is still AI, right? You can talk about all the previous bioinformatics tools that have been generated. Fine. But I think the best tool that we have, the best thing we have in our arsenal as humans is AI. Okay. So now, as a segue, right? HackBio started in the midst of just before ChatGPT was commercialized and after COVID-19 started. So actually in 2020. So this was when HackBio started. And I like to use this screenshot because I like to, I think it's, I've forgotten the title of the movie, of the series, but yeah, maybe those I remember. But this is the professor basically, and it's Money Heist, exactly. So he was introducing, and I'm trying to say, this is where HackBio started, and this is why we started. So we are in the business of training the people, okay? And to a very large extent, HackBio is a little bit differentiated because of the kind of the way we implement our training, in the sense of, I mean, I will still talk about HackBio later on, but I'm not trying to sell on you. But what I'm trying to say is that we come from a hands-on technical perspective, right? You have to do it. Yeah, maybe you need it for something, you need it rushed, fine. But we come from a technical perspective. We are not just learning, but we are making sure you are also solving a major problem in life science as a whole. Okay. But we can talk about that later on. And our backstory is basically, I already said it. We came in just after we came in just after COVID-19 and before ChatGPT came on board and before AI became a thing. So we were also crewed at the beginning, and because everybody was sitting at home, so we decided to just organize something, and it was fun in the first year. But yeah, now we've grown, we've evolved, right? And we are also adapting we're also adapting. I'm seeing some very funny comments on the chats. So we're also adapting to the trends that are happening in biology. Since then, new technologies like single cell technology has come on board. I think nanopore sequencing even got better. And AI is really, really a thing now. AI is so massive right now, and a lot of people are into it. Some of the top startups around the world right now are already into it. It's a money-making machine for startups right now. If you have a very solid idea and you push it out there, single cell sequencing plus AI, just tell them you can use it to solve any disease. Also, I don't know if you've heard about AlphaFold, but the company behind it, which is Google DeepMind, they started a company called Isomorphic Labs, highly funded, and they're trying to solve all the diseases of the world. Yeah, but that's an ambitious project. I mean, Bill Clinton said that about 25 years ago, but still we are nowhere close. But yeah, what I'm saying is because this looks like, so when the sequencing was done, it looked like the best thing after sliced bread in biology. There was no better tool then. Maybe better tools will come, fine. But right now, this is the hottest kid in town, right? And even though there will be more to come in the future, many of the things we would have now, that would come in the future will be based on this. So most of your foundational knowledge has to um, most of your foundational knowledge has to be built on top of this, right? So I will quickly now go into how to start using AI in your research. I hope I've not exceeded my time. Yeah, I'm close to exceeding it. How to use AI in your research. I quickly collected a few ones that I use on a daily basis. So basically, if you want to generate ideas, Claude, ChatGPT, they are good. I mean, Claude is better for coding. ChatGPT, ideas in general. Grok, I personally like Grok because it's very good for writing. If you're writing, it's just a cheat code. If you're writing essays, if you're writing motivation letter, I've used it and I got two prestigious awards recently. So Grok is important. I mean, if you want to use it for writing, it can basically filter down whatever you need to filter down. So Grok is good. Future House, I think they are now highly commercialized. I don't know what happened to them, but you can generate massive ideas. You can conduct literature review on this tool, and then I come to literature review as well. Notebook LM is a very good tool. So you can use Notebook LMs. You can dump a lot of resources, papers, journals, articles, videos. You can dump it on Notebook LM and it will generate a podcast out of it for you. It's very fancy. You can generate a 15-minute podcast summarizing everything. It can help you understand. So it basically helps you to think. And then actual research. If you really want to conduct actual research with AI, I still don't believe this myself, but they provide some tools so it's not as if it will conduct the research by itself. It helps you. Let me put it that way. So OpenBio is a platform. I think, for example, it can design primers by itself. You just have to provide the name of the gene. It would even look for the organism, right? It will even look for the sequence by itself, generate the primers for you, give you the conditions, blah blah blah, and everything you need to do. It also generates protocols, I think, for several scientific methods. NVIDIA started this BioNemo platform where there are lots of AI tools in bioinformatics and life science in general. They just dumped it there. I think there are lots for molecular docking, dynamic simulation. I don't know. I mean, they are so massive. Basic phylogenetics analysis, all of these things are already there. So, um, um, also, I don't know if I mentioned it already, AlphaFold. AlphaFold is basically for protein design. So you can generate new proteins from AlphaFold. And then there is DeepDock. Basically, DeepDock is it's a tool for performing docking, but it's literally based on AI, molecular docking. So yeah, I'll just quickly move. I think I'm running out of time. So how about building models for life science? Because all of the ones I mentioned above is from large corporations, large companies, large people, rich people. But how do you also get to this level? How do you start thinking about incorporating either in your research, because it's a thing now, or start incorporating it as your core, as the core of your research, if you're interested in it? So I think there are three main kind of models that are very popular right now in life sciences. Okay, I know. I see I have 10 more minutes. I know. Thank you for notifying me. But I think in my defense, Michael took more time out of my time. So you have to give me extra minutes. Anyways, there are things we call generative models. So this one's generate things. Actually, ChatGPT is also generative. But yeah, we also have AlphaFold. So AlphaFold can generate, based on an input sequence, it generates a 3D structure for you, a 3D protein structure for you. You also have predictive models that predict an outcome, right? It predicts whether it's binding or not binding, something like this, right? And we have foundation models. So these foundation models are models that are built of massive amount of data, huge amount of data. So you have, for example, the entire NCBI can be trained on something, or the entire PubMed can be trained on something, and you are using it to generate outputs. Of course, it can be generative. I think it's mostly generative at most of the time, foundation models. But the idea for foundation models is that once you have it, you can train on top of it. That is, it's like a foundation, and you can add layers of, they call it neural networks. You can add it on top. Basically, it can advance ahead of, or better than the base state, that the base foundation model. And this one was released by Arc Institute in California. It's a fancy institute. I really wish to work there also one day. It's a very well-funded institute, well-organized people. And maybe there's a Nigerian there actually. Maybe, I don't think it's Nigerian, but it's Nigerian-American, I guess. So maybe you can write to him and tell him you want to work there. Fancy. Okay. But yeah, those are some of the applications, but I also have some ideas about some applications that I think are worth implementing AI models for, and I think you get paid big time for this, right? So I think one major one, maybe this one will not give you so much money, but that's MIC prediction, right? If you're in microbiology, you've probably heard about minimum inhibitory concentration. But this thing is so rugged in terms of predicting, because there's a lot of things that influence it. Two similar organisms, if you put them side by side, they can have different outputs. So why is this? So we need AI models to be able to. AI is one tool that can resolve the genome, the transcriptome, the epigenome, blah, 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 the chemical. It combines everything together and gives you a better interpretation of what's happening. Also, drug combinations. So to find drugs is very hard. How about combining already existing drugs in microbiology, in malaria, in cancer, in diabetes, synthetic biology? When you remove two genes, or when you overexpress two genes, what's going to happen, right? At different organisms, right? Yeast, bacteria, what's going to happen? That's another question that you can solve. Fashion and dermatology. I got fascinated by this because I got an invitation from Nivea. I don't know if they produce roll-on or spray, something like that. Nivea. So they are looking for bioinformaticians in Nivea. And Lowell, I don't know if you know this French company, it's called Lowell. They're looking for bioinformaticians. And your work is basically AI, right? You do basic NGS, then you migrate into machine learning and deep learning. And I think that's fascinating. They're also trying to predict toxicity, the best combination of chemicals that you have to put together, which is quite nice also. And then imaging, you generate a lot of radiological images, ultrasound, blah, blah, blah. I don't know. Could be interesting as well. Anyways, I think that's the end of my talk. But again, this is where HackBio comes in. If you want to get started, I didn't get enough time. I'm sorry, I didn't get enough personal time to prepare But I could also tell you that this year, sorry, not this year, next year, our theme is AI and bio, and we already published this calendar publicly. So if you're interested, you can just mark the date and join us when you are ready. So basically, we start with visualization in the year, but afterwards, we are heavily focused on AI, generative AI, AI-enabled drug discovery. We are not building foundation models because that's much more computationally expensive, right? But someday we'll get there, and yeah, single cell multi-omics in the end. We also have quite some competitions coming along. We'll definitely send an invitation to people in Biospherea through Michael. Yeah. And if you have any questions, you can write to me on Twitter, on LinkedIn, or my email. Thanks for having me, everybody. I'm done. Everybody seems quiet.
Wow. Thank you so much, sir. We appreciate you. And this is a very detailed presentation. You started from scratch, like from the beginning. And we'll be taking questions now. So do we have questions? So you can just raise up your hand. You can raise your hands and we take you one after the other. We have a few minutes for the question and answer. Let me see the chat if we have anything. Okay, more of comments that we got. All right. So I think there are no questions. Okay. So Abdul Basit, your hands are raised. Can you unmute and ask?
Yes, thank you so much, Mr. Adewale. I really enjoyed your session.
1 minute, please.
Can you hear me?
Yes, I can hear you.
Okay, so my question is this. I know that AI is the revolution, is the next big thing. And of course, as researchers and biomedical researchers particularly, there's a lot to do. But I want to ask, from your own exposure and your experience, where can you say AI comes in when it comes to public health and public health scientists, excluding surveillance and modeling aspects? I'm talking about public health on the field and all of those things. Can you just enlighten me particularly?
So originally, I'm not a. Should I take all the questions first and respond, or take it and respond immediately? I don't know how this goes.
Okay, to make it faster, I think we can take the next one too, so you answer together.
Okay.
Emmanuel, please unmute and ask. Thank you.
All right, thank you very much for the presentation. I just have a question. So I think at one of the slides you talked about like trying to discriminate generative from predictive AI models that can be applied to solving biological problems. I think, can you shed more light? What type of problem involving, like what type of biological problem would require you to make use of a generative model? What type would require you to make use of a predictive model? Like maybe AlphaFold, I think is predictive. I think you talk about deep talk. First time I'm hearing that. I think you also classify that as predictive, but then we still have generative models. Maybe you could just shed more light on that. I'll be glad. Thank you.
Yeah, thank you. Good questions. Very interesting question. So public health applications of AI. Personally, I'm not into a public health person. I'm not a public health scientist, so I just make a disclaimer. But from someone that is far off, like I'm looking at what has been happening in public health so far, I think public health deserves a foundation model at this time. okay, at this time. So of course, I think majority of the work you do in public health is mostly um, how do you say, forecasting, epidemiology, foundation model that can accurately. So the thing with foundation model now is it can generate a series of events. So here's the thing with foundation models. They are usually encoder, decoder architecture. So basically, it collects everything that has happened in the past, encodes them, like understands what's happening, but then it also understands how event A leads to event B, right? So it's like a network of things, how it happens in space and time. So I think what a foundation model can give you is once you have it, or like a generative model, once you have it, you can bring in the latest set of things that has happened in terms of epidemiological trends, and you can tell it to predict. I know this is also what you do already, right? But maybe much more accurately, AI can do that for you. There are also the thing with stewardship, especially in AMR, communicating the right kind of drugs to use, communicating to people. I think AI can be much more knowledgeable in personalizing these messages to people. I think this is also another interesting aspect of public health. Maybe my ideas are not solid enough, but yeah, these are a few things I think you could try out in AI about generative and predictive and foundation models. So generative, as the name implies, it's to generate. Okay, so if you have an input, you want to be able to generate an output. I didn't follow exactly Michael's presentation, but I think we worked together a few months ago. And the idea that we were trying to do is that if you have a transcript of something that is healthy, okay, or let's say this is an healthy person, or this is a sick person, approximately. Yeah, let's say this is a sick person, right? And then you put a drug. I want to be able to tell the outcome of that drug on the transcriptome without sequencing the person's genome. Okay, so basically what we did when I was working with Michael was we have samples from space and then we have samples on Earth, right? So I think it was an hackathon. It was not the main project. So what we wanted to do was, without going to space, we want to know the effect of the radiation, the microgravity, the everything that happens in space. We want to know the effect on you before you go to space. Whether there is, we just want to know what happens. We want to be able to predict what happens to you before you go to space, even while you are still on Earth. I don't know if it makes sense to you. It's kind of weird, but I think we got some very good correlation, although our data size was small. But yeah, we got some good output, and we were planning to actually write it, but I don't know the status of that right now. Yeah, but basically what I'm saying is that's generative model. So it's going to generate an output for you based on the set of inputs or the query you give to it without ever seeing it before now. Now, for predictive, so generative is also AlphaFold because generative, you're going to just supply, for AlphaFold, you're just going to supply your primary amino acid sequence and it generates a 3D structure. Not seen that 3D structure before now, but based on the primary sequence that it was trained on, it can generate a 3D structure. So, but predictive model is you just want to get a regressive value. So, for example, you have 2, you just want to get 2, you just want to get 3, you just want to get 34. Either it's drug concentration, it's plant height, that's the predictive step. But the foundation model is, I think most of the foundation models out there are generative, right? They are generative in the sense that they also generate outputs. But the fun fact with foundation models is that they are just foundational. Of course, you can use them as it is, But of course, but again, you can build on top of it. You can build on top of it. You can build new things on top of it. You can use it as a starting point for another neural network. So you don't have to use 96 million GPUs that they used. You just have to plug it into your own personal computer and then build on top of it. I hope I answered the two questions.
Yes, you did. Thank you so much, sir. We really appreciate you. So we've come to the end of this session. Can we appreciate him? Can we drop in some emojis to show how much you've been impacted by this session? Yes, I also learned new things even about the tools, AI tools. Some I've not heard of them before, so I'll be trying them out. All right, so we'll be moving to the next session, right? And before we move to the next session, I just want us to quickly drop it in the chat. You know, we've been talking about AI and the like. So what in one sentence, how would you describe your relationship with AI? Can you just drop it and let's see. Are you curious? Are you skeptical? Are you excited? Are you confused? So let's know. How do you feel about AI in science? You can keep your comments rolling. Did you get my question? Your personal assistant. Yeah. Okay. Any other person? Let's keep that rolling. Right. On the fence. Yeah, on the fence. So that's being skeptical, very distant. Like you're trekking from a long distance, right? You're curious, excited about the future. Yeah, it's exciting. Yeah. Comfortable. Fingers crossed from Martin. All right. Thank you, work buddy. Mm-hmm. So AI can be many things to you. And so let's see how it unfolds. So we'll be moving to the next session. We'll be moving to the next session, and that will be the poster presentation by Babalola Blessing. And she'll be presenting on bridging the diagnostic gap, protein biomarkers for rapid schistosomiasis in resource-limited settings. So this presentation is a collaborative work she did with other people. We have Mercy, we have Kabirat A, Sulaiman Tajuddin, Oriade, Oyetunde T, and Oyeyemi. And Babalola Blessing is from the Department of Biosciences and Biotechnology, the University of Medical Sciences in Ondo City. Let's give a round of applause or put in some fire emojis as Blessing comes up. Babalola Blessing comes up to present.
Good evening, everybody.
Good evening.
Can we hear me? Okay, yes. My name is Blessing Babalola, as introduced. A minute, please. I want to share my slide so we can see. It's gonna work. Okay, can we all see my screen, please?
Yes.
All right, a minute, please. I just want to make it as visible as possible. Okay, my name is Blessing Babalola, and I'm here to make a poster presentation on the topic Bridging the Diagnostic Gap, protein biomarkers for rapid schistosomiasis detection in resource-limited setting. First off, schistosomiasis is a parasitic disease that is caused by schistosomal flatworms. It affects closely 250 million people globally, of which sub-Saharan Africa it's the hardest. So the current diagnostic approaches that we have often faces challenges related to sensitivity and specificity. Although these approaches they are plausible, but the heterogeneity, one of it is crude parasite antigen, and the heterogeneity of this parasitic of this parasite antigen compromises test specificity. So which calls for a more reliable diagnostic measure which I'm suggesting, that is elicited schistosomal proteins, which can show promise for rapid field diagnosis. So the aim of this study is to validate high-performance immunogenic proteins that were gotten from schistosoma soluble egg antigen and worm antigen protein. Now, my research question: do specific immunoreactive protein bands identified from schistosoma mansoni, SIA, and WAP demonstrate clear antigen antibody recognition on dot blot platforms for schistosoma hematobiome diagnosis? Simple put, the protein bands that are going to be gotten from these eluted antigens, can they demonstrate antigen antibody recognition when expressed on a dot blot platform? So my materials and method: 20 urine samples each of schistosoma hematobiome and positive and negative were utilized. So this is my workflow. Western blots was used to identify the immunoreactive protein bands that are present, while STS page was used to the identified immunoreactive protein bands from Western blot was eluted in cut out. The bands were expressed according to their molecular weight. They were cut out. Afterwards, they were estimated. Protein estimation was done. Next on indirect ELISA workflow. The indirect ELISA is to quantify the already identified immunoreactive protein bands. And dot blot was done to validate the immunoreactive protein bands. And then statistical analysis was carried out. The area under the ROC curve. So there are different grades for area under the ROC curve. They can be 0.5, shows that there's no discrimination. Area under the ROC curve that has 0.7 to 0.8 shows that it is acceptable, while 0.8 to 0.9 shows that it is excellent, while 0.9 to 1.0 shows an outstanding result. Now, these are my results. From my methodology, I said Western blot is going to be carried out to identify, to point out the immunoreactive protein bands. So in this image, we can see an arrow that is pointing to a particular band. That's band 43 for SMCR, and this arrow is pointing to band 55 for SMWAP. So this image is for the Western blot. These are the bands that we expressed, and this is the image for the SDS page. So it was on this SDS page, this is a gel, it was on this gel that this band was excised. Also on this gel that this band was excised. Afterwards, ELISA was carried out with those bands. They were crushed and we got the protein and I used it to carry out ELISA. So this is a table that is showing the performance of this eluted antigen, not the crude form now, the eluted one, because they've been expressed and they've been eluted. So having sensitivity of 95%, and specificity of 95%. So in diagnostics, sensitivity just means the ability of a diagnostic tool to tell positive candidate of a particular disease, while specificity tells the ability of a diagnostic tool to identify negative results. So this particular, and the higher the area under the ROC curve, the more sensitive the more sensitive the diagnostic tool is. So the sensitivity for this particular eluted band was 95%, and the specificity was also 95%, while we have the accuracy of 95% and 92%. This table is also showing our true positive to false negative and our true negative to false positive. Now, this is my ROC curve. So in a ROC curve analysis, the more this arrow tilts to the left, the better the diagnostic precision. And as you can see in this particular image, the ROC curve is tilting so much to the left, showing the diagnostic potential of both eluted bands. The area under the ROC curve is 0.95, and the area under the ROC curve is 0.95. Then going back to the statistical analysis, any area under the ROC curve that falls underneath 0.92, 1.0 stands for an outstanding result. And that's what was recorded. They had area under the ROC curve of 0.9%, 0.95 for both, and also the p-value 0.0001. This means that the tendency that the results gotten from this is random is 0.001. While my second figure is showing the antichistosomal IgG production. So I worked with 20 urine samples, positive and negative. So this is the negative. The antibody titer that was being expressed in each of the urine is recorded in the blue, while the antibody titer that was recorded in each of the urine samples that are positive is recorded. So looking at the positive to the negative, you can see the antibodies that were being expressed as in comparison to each other. While this is the dot plot, my research question, simply put, asked if this eluted immunoreactive band can identify the antigen to antibody interaction on dot blood platforms. So this is schistosomal mansionii serum for band 43 showing the positive result. These are distinct results that shows that this particular immunoreactive band can identify positive samples, positive urine samples. And this is the negative dot blood platform. There was no reaction that went on at all. And for the second immunoreactive protein band, that's SM45, they were able to identify positive distinctly because this is very pigmented. And the negative, the PVDF membrane, the dot blood platform remained clear. So the implication of my study, different immunoreactive protein bands were identified through SDS page. I can't find my screen. Okay, sorry about that. Different immunoreactive protein bands were identified through SDS page and Western blot, among which SM-CR43 KDA and SM-CR55 KDA demonstrated outstanding diagnostic performances. And also, ELISA analysis reviewed the high diagnostic accuracy, confirming that these antigens have the ability to detect schistosomal-specific antibodies in human urine samples, while dot blood validation furtherly confirmed their immunogenicity, which produced visual differentiation between the positive and the negative samples. This is underscoring their potential application in field diagnosis for schistosomiasis. In conclusion, SM, schistosomal mansionii 43 KDA and schistosomal mansionii 55 KDA proteins show strong diagnostic performance, supporting their potential for rapid schistosomiasis detection in endemic regions. This are my references. This is my reference, just one. Yes. Thank you so much for listening.
Thank you so much. Thank you. Well done. This was a beautiful presentation and very succinct, straight to the point and right on time. Okay, so do we have questions for her? Do we have questions for Blessing?
What method was used for protein extraction from the gel? Maybe we could take that.
Protein extraction from the gel. Did I get that correctly?
Yes, please. Yes, please.
Okay, so I couldn't do too much explanation because of the time. So let me just run the SDS page workflow so you understand how it was extracted. So the gel was it was separated according to their molecular weight. So the elution is just using a sharp blade to cut out the required or the target bands. So after cutting them out, excising them, they were crushed. This image, like we took out the necessary bands, crushed it using a homogenizer, then we centrifuged it. So now the supernatant was our antigen. That's what was made use of. Afterward, it was estimated. So simple put, the method that was used to extract was just excising the bands, crushing it, centrifuging it to get our antigen and estimation. Did I answer the question in a way that you understand?
Yes, I think you did justice to that question. There are so many questions on the chat. Maybe Blessings can answer them directly because time is not on our side.
Okay, I'll
Yes,
do that.
yeah, thank you, Blessings. Please interact with her on the chat.
Okay, I can stop sharing my screen right now.
Yes, thank you.
Thank you. Thank you too.
All right. Thank you so much, Adede, for stepping in. My apologies, I had a little technical glitch here. So we've come to the end of today's session, and I want to thank everyone. Thank you everyone for being a part of this celebration. Today is day one, and to every speaker, every presenter, our contributors, and every member of this community, we want to say a very big thank you. And today is a very rich reminder of why Biospherea exists, which is to create a space where African bioscientists and even those in diaspora can learn, we can grow, collaborate, and also lead meaningful conversations that shape the future of science. And really from the keynote presentation, even down to the poster presentation that we just had, we add diversity of insight that were shared today, and that reflects the strength of this community. And to our speakers and presenters, thank you so much for your time. Thank you for your generosity. To our audience, we say thank you for engaging and asking thoughtful questions. We really appreciate that. I hope today you were inspired, you were encouraged and connected. And tomorrow we still continue this session, and we would like to invite you to join us again for day 2 by 8 p.m. Watt. And you can convert that to your time zone. We have another exciting lineup of, activities, sessions that will be filled with deeper insights, and you don't want to miss that. You can also invite your friends, invite other people to be part of this. We'll be having more conversations and even more opportunities to grow together as a community. And also, I would like to like us to know that this meeting is recorded, so it will be shared with everyone. You can always go back for a recap and just enjoy all over again. And so finally, on behalf of the organizing team and the entire Biospherea family, we want to say thank you for spending your time with us today. Have a wonderful rest of your day, and we look forward to seeing you tomorrow. Thank you so much, everyone. And you can just drop something in the comments to say, just give one word for today's session, today's meeting. What was it like for you? And we end today's meeting. Yes, Augusta said knowledge filled. I agree with that. I learned a lot of new things. Ruth said smooth. Thank you. Captivating. Elite. Yes. Okay. We can have more comments rolling in. Beautiful event, inspiring. All right, wanting more. So if you want more, we would like you to join tomorrow again. And do not miss it. Thank you so much, everyone. Encouraging, yes. Uplifting, refreshing, enlightening. So it's a whole lot. It's a whole lot. Yes, interactive and informative. I agree with that. All right. So as Martin said, he said if we are related, we'll meet again. So let's see you again tomorrow.